So I'm sure that everyone that's ever solved a crystal structure has complained about the amount of metadata that they've had to put in when they deposit in the PDB, but we also know how important capturing this metadata is for kind of establishing databases that are able to give us programs like AlphaFold. So it's my pleasure to introduce Ed Daniel from, okay, my Swedish is getting better, my Finnish is still pretty bad, who will be talking about LIMS systems in crystallography.
Well, that was the intent anyway. Yeah, should have known better than to try and get the presenter view up, shouldn't I? There we are, that'll do it. All right, good morning, and thank you very much to the organizers for giving me the opportunity to present here. So, first question, apart from how do you pronounce Oulu, because I still haven't figured it out after 12 years, is, where is it? Why is this not working? So I was just barely on the edge of Nick's map, and just had one thing to say, 6 hours of daylight? Bless. So this was noon on Monday, and the only reason I can see the sun is because there's a gap in the trees. So Oulu is famous for two things. First of all, being a year-round cycling city, they're very hot on keeping the infrastructure going. My head of department stops cycling when it gets down to minus 30 degrees, but only because the grease in the bearings makes it too hard to pedal. And the other thing we're famous for is the Air Guitar World Championships, which is every bit as silly as it sounds. Yeah, come and see us sometimes. It's a fun place. All right, so down to business. We're going to talk about the overall big picture of why this stuff is important, and then we'll take a look at LIMS versus ELN versus paper notebook, advantages, disadvantages, and all that good stuff. And then we will step through that whole workflow with IceBear, and hopefully we'll see how we can accomplish the goals that we have. All right. So yeah, the general workflow, you're almost certainly familiar with it for the routine stuff. Do a whole bunch of trials, get some crystals, ship it off to a synchrotron, shoot it, solve the structure, deposit it. But that's no use unless we have this complete chain of custody where we know the protein sequence that we started with, how we got the crystals, how we got the data, what we did with the data, and how it was deposited and all linked back together. Seems easy enough, right? Well, not quite. Depending on what you're working with, I mean, ask the poor membrane people, protein people, you could be looking at years for your crystallization trials. Then you get to the point where you have a crystal and suddenly it's the opposite problem. If your place is anything like ours, then DHL are picking up the dewar in 2 hours and people are fighting over microscopes and madly trying to fish, and there's hundreds of crystals everywhere and it's chaos. Then you ship it. With any luck, DHL don't lose your dewar. Ask me about that later. You get it to the synchrotron, and suddenly you might have terabytes of data coming from your crystals. What do you do with it? Then you get it home. Maybe you don't even get it home yet. You try and solve the structure. It could be a couple of minutes, but again, it could be years. And now we have a problem where the person who started the work may well not be the person who finishes the work, and somehow we need to do some kind of handover. And assuming we get to the end of the process and we want to deposit and publish, more and more funding bodies and some of the journals are requiring that the raw data is available. More and more we're hearing about this FAIR data, which I always forget what it stands for, but it's important. That much I do know. So the only hope that we've got, if we're going to get to the end of this process, several years after we started, and still have everything that we need, is to get organized. And chances are the paper lab notebook is not going to cut it. So let's use a computer. That always makes things better, right? So a couple of options here, and I honestly don't think that there's necessarily a a sharp line that you can draw between these two, but the electronic lab notebook is trying to keep some of the flexibility of the paper notebook, but imposing some structure. And you may be able to like set up a standard form for inputting an experiment to give you some kind of structure, but you are more able to change that. And you may have some integration with lab hardware, whereas the LIMS is more the more heavy duty version of that, where you know exactly what you're doing it over and over again. It's much more optimized to a given task, and there's probably less flexibility within the system, and hence a somewhat steeper learning curve in many cases. The payoff for that is that it's typically integrated with a lot more lab hardware. So instead of having to fill the information in, the information will be filled in for you. You do the science and it just ends up in the database, which is really the ideal situation. As I say, some well-featured ELNs might start to look like a LIMS. Some less featured LIMS might start to look like an ELN. And what you call it, well, may well depend on the marketing department. Of course, being broadly similar in what they do, they share a number of benefits. Obviously, being able to link with one experiment to the next, to the next, to the next is a big one. It's kind of hard to do with a paper notebook. It's much more collaborative than the paper notebook. We talk about access control, which is kind of strange because we're actually giving more access. We're controlling how we give that access, but when you have everything on a computer and you can log in through your web browser, more people are able to see the data, and more people are able to collaborate. We mentioned already the possibility of the project handover. Depending on how that's done in your system, that could be a 10-second job. Just change the owner of a project and the new person can then walk in and take over. Adding and attaching files, it's always a good fallback option if the software doesn't support exactly what you want to do very well. Again, try that with a paper notebook. And we mentioned the lab hardware integration. That really is key. And as we saw in the last talk, the ability to search these databases and get things out is really quite important. Of course, nothing in life is for free. And yeah, to err is human. To really screw things up, you need a computer. So yeah, you're putting all this stuff in a database and you're hoping you don't get hacked. You have to trust that there are backups. You do have backups, right? I've saved at least one PhD thesis where the guy came to my door with a dead laptop, and he thought he might have put his thesis on a stick 6 months ago, but he wasn't sure where the stick was. So, you really have to be on top of this stuff. The one that keeps me awake at night as a developer is that weird software bug where it looks like the data is going in okay, and either it's actually getting slightly corrupted or I'm just completely dropping it on the floor. And it's not until a year later when you want to go back in and find it that you realize there's a problem. I can mitigate that to a certain extent with decent software development practices, but yeah, that's the one that keeps me awake at night. In terms of the security and the data loss, you can mitigate to this to some extent by outsourcing it. If the LIMS or ELN provider lets you host it at their infrastructure, then at the very least, you've got somebody you can sue. Okay, so which is best for what you're doing? Well, depends what you're doing, doesn't it? But you know, you can't beat the lab notebook for the paper notebook for the freedom to just scribble ideas. And the ELN tries to give you as much of that as you possibly can get, but it does impose that bit more structure. Whereas the LIMS is great for the highly optimized. You know how you're doing the process. You've got it so optimized. You've automated it, and you're just cranking stuff through and letting the machines put the data into the database for you, and that's fantastic. But no one of these may be the right answer. I know in our place we have at least one group where they all the plates go into the LIMS, but they also use an ELN, and they put the what in the LIMS and the Y in the ELN, and then they link between the two of each other, and you'll still see them scribbling on paper as well. So there's no one perfect solution. However, I'm going to assume for the purposes of this talk that the LIMS is the right answer. Now, nothing to do with me being a LIMS developer whatsoever. I'm not at all biased here. But for the routine stuff, it does really come close to that, that model where we know what the process is and it doesn't really change. So then it comes down to which LIMS do you use. If you've got automated imaging hardware, there's a very good chance it's Formulatrix, and if it's Formulatrix, there's a very good chance you're running RockMaker. They will also allow you to drive dispensing robots through that, and they've got some really nice experiment design tools. I'll be honest, I'm a little bit scared of it. It does so much that I sit down in front of it and I don't quite know where to start sometimes. CRIMS was mentioned in the last talk. That the guys at EMBL have done a really, really good job with the hardware integration. They will also drive Formulatrix imagers, just completely replace RockMaker. Automated crystal harvesting, all of that good stuff, they can do that. And their software can submit shipments to ISPyB as well. And then there is IceBear, which I'm most familiar with, so that's what we're going to be going with for the rest of this talk. And we sort of cover the whole pipeline, a big asterisk there. And we've put a lot of effort into integrating with the ISPyB synchrotrons for exchanging shipment data and auto-processing data, which we'll see later on. So best place to start with IceBear is a project. This is sort of the smallest thing that we can apply access control to. So that will contain all the work on a given protein. Typically, I would recommend having one protein per project. That means it's easy to apply the right level of access controls. There's a project owner who is able to manage the proteins and the sequences and do some other project-specific tasks. And then we grant access to given user groups. So for an external collaborator, we might give them a read-only access so they can see what we're doing. Again, the access control gives you the security, but it also makes the whole database feel a lot smaller, because 90% of what's in there, you're never going to see it. You're only seeing your own stuff because of the access rights. Handover we mentioned, just go into the software, 10 seconds, change the owner, job done. And at the end of the process, for now, we can grant read-only access to the project for a guest account, and that sort of makes it public. It's not ideal, but it's a start. But we also integrate with imagers. So we pull in the imagers and the plate metadata from our Formulatrix system. And as you can see, we can play the images one after another and see this fantastic crystal grow. But at the same time, we're also gathering the incubation conditions, what plate type, and all the drop volumes. That's all attached to the protein sequence. So we're already starting to build up our chain even at this point. So we found a nice fat crystal, and now we want to go and do something with it. So obviously we move into crystal fishing. You can see here, Christian is at our fishing station, and on his right-hand side there, he has this crystal fishing interface. We've designed this to be as hands-off as possible, while still allowing you to do the IT and the science at the same time for the minimum level of confusion. So this is a nice drag-drop interface. We have a touch screen here. We have a barcode reader, so we tend to just scan in our pins and pucks and dewars before we start. Scan in the plate. You can then drag from the crystal, drop it on the pin. Crystal goes into the pin, pin goes into the puck, and then when the puck's full, you can put it into the dewar. So there's a bit of a blurry line here between shipment assembly and crystal fishing. It all kind of happens at the same time. And yeah, as I say, in our case, it usually happens in the last couple of hours before DHL show up. But this reduces the possibility of getting the wrong crystal associated with the wrong pin on your paper, because you're just done. When we actually get round to shipping, here's our dewar with our sample in it. We click send shipment. We then authenticate with our synchrotron credentials, and then we choose our proposal and session. We choose a lab contact from the ones that are already on the proposal. If it's diamond, we will have to choose a safety level for the shipment. We then do a quick review, make sure everything's cool, hit the button, and the shipment is sent. And we've now sent all the details of our crystals in the shipment. The only thing we typed was our username and password for the synchrotron. It's just all gone. So now you can see that the shipment is in ISPyB with all the details that we filled in. There's our dewar, and you can see our sample. So no typing, just done. If you've ever had to fill in the spreadsheet and put a shipment in that way, I think you maybe appreciate how cool that is. But we've also swapped URLs. So there's a crystal URL for IceBear. The crystal will have a URL in ISPyB. IceBear and ISPyB now both know about each other. So in theory, at least, it's possible to just click backwards and forwards between the two and see your crystal in both systems. Then we come to data collection. The way we tend to do this here in Aolo rather is we have a big screen with the ISPyB and Beamline stuff on it and collecting the data. And then on the smaller screen, we have IceBear with a separate keyboard, and we can just rattle in notes about the data collection as we're doing it. We're also deciding at this point, do we want to wash the pin when it comes back, or do I, for some reason, want to keep the crystal and send it somewhere else again? And all of that is, again, being attached to the crystal. So we're still building up this chain. Once it's all done, we hit the retrieve metadata button, again, authenticate with our synchrotron password. Everything gets sucked in and attached to the crystal. We pull in all the data sets, attach them to the relevant crystals. We pull in all the auto-processing pipeline results, attach those to the relevant data sets. We're also capturing the location of the raw data so that we can find it again later. And we're also getting a snapshot of the detector information. So when we come to deposit 5 years from now and the beamline has been upgraded, we still have the original and correct information there. And we can then get a report of everything that happened in the shipment. We can sort it by resolution or whatever we feel like. Okay, then magic happens. You solve the structure. You may notice that I'm somewhat glossing over this. Then we come to deposit it, and we can now associate the PDB code with the individual data sets and with the crystals as well. And so now we've got a pretty much complete chain of custody from the protein sequence all the way right through to the PDB code. And from IceBear, you can obviously click off into the PDB and go look at the sequence, the structure even. All right, so broadly speaking, we managed to do everything we wanted to do. We've brought our images and our crystallization data in from the imaging system. We've fished the crystals. We've managed to get some data and attach that to the crystals. And we've done magic, and we've got a PDB code. Everything's in there. It's archived. Assuming our IT are up to scratch, it's backed up, and we can share the project with anyone else that we need to do simply by giving that guest user the read-only access. So that's it in terms of sort of LIMS in general and where IceBear is at the moment. In terms of future work on IceBear itself, I don't really like the process of linking the PDB codes in yet. It's still a little bit of a faff trying to find them all and do it manually there. I think there's a better way of doing it, but I just quite haven't figured that out yet. I'm well aware of the irony of standing here at the CCP4 study weekend and having completely glossed over the structure solution part of the process. As with anything in IceBear, you can always attach files to the relevant crystals or data sets. but there has to be a better way of doing that, and that's very much on the radar. Also, making a project public is not really fulfilling the fair data criteria, so we're going to put some work in that. Managed hosting, we mentioned earlier. So we host the Finnish IceBear instances on the CSC cloud in Finland, but for a number of reasons, not least of which is the complication of billing outside of Finland, we are looking at bringing that hosting in-house. So if you wanted to run an IceBear instance, then we can host it at Oulu for you, and we can even plumb it into your images remotely if need be. One thing I never expected to have to integrate into IceBear was GPS trackers, but DHL. We've lost viewers. We've had viewers show up months later. We've had viewers not show up. So James Hall at Diamond put me onto these Lightbug trackers, and they have GPS and cellular on board, and you just pop it in with the viewer, and you can see exactly where it is. And yeah, it's fascinating. DHL say the viewer is in Paris. Diamond say the viewer is at Diamond. The GPS says it's at Diamond. So it's a flight and two vans between the two data points. But the other cool thing about the Lightbug is that it has a really nice software API. So we can start to trigger actions when the viewer arrives at the synchrotron or when it leaves. And you can change the UI in IceBear, or send people emails to say, hey, the viewer's back and you need to clean your pins. All of that sort of fun stuff is on the horizon. I'll leave it there. So thank you again to the organizers. Thank you for paying attention, or for not paying attention, but not asking any awkward questions. It's all good. Thanks particularly to the ISPyB developers, without whom I couldn't have done any of that integration, and also to the IceBear user community who do ask a lot of awkward questions. And finally, to the kind people along the bottom who pay the bills. Thanks.
Right, are there any questions? Sam, if you could run off, I will run up to join you.
Am I going first? Okay. Hi, Samuel Foster, University at Leicester. Thank you for the talk. I was curious, when you're fishing your crystals and putting it into the LIMS, is there a way to note that you use a cryoprotectant at the same time? Because it didn't appear. Yes,
it does. It does allow you to do that. I haven't actually shown it here, but once you've done the fishing action, by default, a box pops up with space for you to write notes in. There are some buttons there, so you could touch quickly to add sort of common notes without any typing, and that goes away if you haven't done anything to it within a couple of seconds. Personally, I find it annoying, turned it off, and I've completely forgotten the option exists. So thank you for reminding me.
And a quick follow-up, if I'm allowed to. This process obviously gives you provenance for your data for years and potentially. What happens when IceBear updates? So if you go to, say, Polar Bear, your follow-up program, what happens with your data if it's going to be incompatible?
Yep, that's always a question. It's always a risk with putting these things into these systems. I would say that this is for the long term. We've gone through several versions of IceBear, if you like, and the update for that is having an administrator go into a relevant section and press a button, and everything just keeps updating. But again, this is part of these fair credentials, which, as I say, I always forget what they are, but there. So the ability to get the information out of IceBear in a standardized format that some other LIMS could eat if for some reason IceBear stopped being developed or supported.
Cool. Thank you.
Need to unmute.
I can't hear you.
Andrew Perkins, Francis Crick Institute.
Hi.
We recently bought a crystal shifter.
Oh, nice.
Is there options for integrating that sort of data into IceBear?
Not at the moment. I would need to take a look at the format. I know CRIMS does support that one. So if you're going down that road, you might want to talk to those guys.
Yeah.
But by all means, I'm happy to have a look at it.
Happy to
And
talk with you maybe at lunch.
absolutely.
Okay, thank you.
Oh, this is.
Yeah, this is the shift now.
Sorry, I saw who it was.
Nathan Wright, Oxford University. I did the shifter, so I'm sorry. But it's all done. It's all stored. The data's all stored in a CSV, so you can pass it out. Okay, but
Right. So I guess if they just had a shared drive, I could pull that CSV in and parse it. Yeah, just keep doing that over. Yeah, sorted.
So that'll be done in a couple of weeks. Question from James Holton in Slack. So I remember getting pushback from shipping companies on putting battery operated trackers in our dewars. Have things changed? Or does DHL just not know?
DHL don't know, but I did look up the IATA dangerous goods regulations, and the batteries in these light bulbs are smaller than the maximum. So there is no paperwork required. And if they whine about it, then you slap them around the head and you point them at the DG regulations.
DHL are actually on their website as a recognised partner.
So Livebug have got DHL as a recognised partner on their website. So
That's nice to know. Thank you.
Got a quick one. If you're upgrading to polar bear, will your DNA on your logo not be symmetrical anymore?
Hey, I'm not even going to comment on the logo. We needed something in a hurry to prevent my professor from calling it something with lots of X's and 2's in it. So we came up with that in an afternoon. Yeah.
Nebaderi King's College London. Just want to find a quick one. Is this open source LIMS?
Yes, it is. It's MIT licensed.
And I guess a secondary question to that from me, how hard is it to set up in your lab from scratch? If you say you can connect remotely, fantastic, but like realistically, how much effort's required?
Okay, so I designed it to be installed by some poor postdoc who came to a meeting like, or whose professor came to a meeting like this and said it would be really nice if this was working by Saturday. Been in that position before. So yeah, you need a box with Linux and Apache, MySQL, PHP, and then you download the tar file from the website. You run a script, and then you log in on the website and walk through some questions. So it should be fairly simple to install. I would say that I think I've broken the installer in the latest release. So if you are looking at installing this to play around with, then I would install version 1.7 and update from there. But come and have a chat with me or fill in the contact form on the website.
Emily Cross asks, what's the plan for raw data / images? Will that be made accessible as part of FAIR data?
Yeah, this is a difficult one. Do you want to pull all these things in from the synchrotron only to then store them locally and then stuff them into cold storage somewhere else? So now we have 3 copies of these terabytes. It's a really messy question. I would say that because we have a link to the raw data and the raw data is at the synchrotron, then if it's made available there, then that's probably good enough that we can just go from the public project in IceBear, click the link and go straight to the raw data.
So it's specifically for Diamond, I guess, because it goes onto tape eventually. Is there a way that IceBear might be able to talk with ICAT to get it off tape?
Quite possibly. These are questions that we haven't had to deal with yet, and that tends to be the driver, to be honest, is when we hit a wall we start developing. But more and more we are copying the raw data from the synchrotron into cold storage at CSC, where it doesn't cost us anything, and we're updating the raw data location as we do that. Might be nice if we can do that automatically, press a button, have it sucked in and squished out and update the location at the same time, but that's probably as far as it will go.
Okay, great, and thanks. I think in the interest of time, well, we're perfectly on time, so we'll move on to the next speaker, but thank you very much.
Thank you.